Welcome friends to a new module of application of Perl to bioinformatics. Here we discuss the remaining part of the genetic code. The genetic code part 2. As we have seen earlier, the genetic code is how a cell translates the information contained in its DNA into amino acids and then proteins. DNA encodes the primary structure of proteins. DNA has 4 nucleotides, namely adenine, guanine, cytosine and thymine, which are commonly referred with one-letter abbreviations as A is for adenine, G is for guanine, C is for cytosine and T is for thymine. For computational purpose, a strand of DNA can be represented by a string of A's, C's, G's and T's. Likewise, proteins have 20 amino acids. The encodings work by taking each group of 3 nucleotides from DNA and translating them to an amino acid or a stop signal. The group of 3 nucleotides is called a codon. In the following sections, we are going to see how this coding and translation works. Coding and translation. Transcription is the first step of gene expansion. First, transcription uses DNA to make RNA and then translation uses RNA to make proteins. This is called the central dogma of molecular biology. It is the explanation of the flow of genetic information within a biological system. Or in other words, it can be described as DNA makes RNA and RNA makes protein. During transcription, a DNA sequence is read by an RNA polymerase which produces a complementary antiparallel RNA strand called a primary transcript. In translation, messenger RNA or mRNA produced by transcription from DNA is decoded by a ribosome to produce a specific amino acid chain or polypeptide. The polypeptide later folds into an active protein and performs its functions in the cell. The whole process is easier to simulate on computers using strings to represent the DNA, RNA and proteins. Transcribing DNA to RNA is very easy in computer simulations as it is just a matter of changing one letter to another, keeping in mind that the actual process in the cell is much more complex. So, we may now skip the transcription and concentrate on translation. With 4 kinds of bases, each group of 3 bases of DNA can represent as many as 4 into 4 into 4, that is equal to 64 possible amino acids. Since there are only 20 amino acids plus a stop signal, the genetic code has evolved some redundancy so that some amino acids are represented by more than one codon. This chart shows how the various bases combine to form amino acids. An important characteristic to notice is redundancy, that is, the way more than one codon translate to the same amino acids. The machinery of the cell actually starts at some point along the RNA and reads the sequences codon after codon, attaching the encoded amino acid to the end of the growing protein sequence. Translating codons to amino acids. The first task is to enable the following proteins to do the translation from the 3 nucleotide codons to the amino acids. The most important step in implementing the genetic code is encoding the amino acids by 3 nucleotide codons. A subroutine to return an amino acid from a given 3-letter DNA codon can be designed like this. We will call this subroutine codon TAA. Here the subroutine is to translate a DNA 3-character codon to an amino acid. Here each of the if statement compares the codon to the regular expression. It can be specified as sub codon TAA. First of all, the argument is collected and is initialized to the variable codon. And the codon is compared within the if statement to the regular expression starting with tilde symbol. If it is a match, it will return the first letter of the corresponding amino acid. If it is not a match, it will go to the next else if statement which compares with another amino acid. This process continues until it finds a match, or else it returns an error message which is given as it's a bad codon. Here the error message uses the standard error file handler called stderr. There are three standard file handlers: stdin to read the user input from the keyboard, stdout directs output to the screen or another standard place, and the stderr for standard error messages which are usually printed on the screen. Though it takes a lot of string comparisons, the code achieves its purpose. The redundancy of the genetic code. We have already mentioned that the genetic code is redundant. From the previous subroutine, it can be noticed that redundant codons almost always have the same first and second bases and vary in the third. Now we will rewrite the subroutine to make one test for each redundant group of codons. The subroutine codon TAA. It is a subroutine to translate a DNA 3-character codon to an amino acid. Here also the arguments are collected and initialized to the variable codon. In the first if statement, the codon is compared with the regular expression, in which the regular expression is specified with only the 2 characters of the codon and the third letter is specified as a dot. This dot matches for any character of the amino acid. It will return an A which stands for alanine. If it is not matching, it will go to the next else if statement, in which the codon is compared with the next regular expression. Here the regular expression is specified with 2 characters of the amino acid and the third character as a character class. This process continues until a match is found. If the specified codon is not matching with any of the regular expression, it will go to the else part at the end of the program. It is a print message to print bad codon. Using character classes and regular expression, this code shows the redundancy of the genetic code. Here the 1 character codes for the amino acids are now in alphabetical order. A character class such as TC matches a single character, either T or C. The dot is the regular expression that matches any character except a new line. The GT./I expression for valine matches GTA, GTC, GTG and GTT, all of which are codons for valine. The I after the regular expression means match uppercase or lowercase. For instance, /T/I matches capital T or small t. The new feature in this regular expression is the use of the vertical bar or pipe to separate 2 choices. Thus, for serine /TC./AG, the character class TC /matches /TC dot or /AG with the character class TC/. In this program, only 2 choices per regular expression is necessary, but we can use as many vertical bars as we click. It is also possible to group parts of regular expression in the parentheses and use vertical bars in them. For example, /give me a pen or chalk /matches give me a pen or give me a chalk. Since Perl is highly optimized for string based operations, working with DNA sequences will be extremely fast in Perl. Now we can consider some of the salient features of genetic code. 1. The codon is triplet. 61 codons code for amino acids and 3 codons do not code for any amino acids, hence they function as stop codons. 2. One codon codes for only one amino acid, hence it is unambiguous and specific. 3. Some amino acids are coded by more than one codon, hence the code is degenerate. 4. The codon is read in mRNA in a contiguous fashion. There are no punctuations. 5th one, the code is nearly universal. For example, from bacteria to human, UUU would code for phenylalanine or Phe. There are some exceptions to this rule. Sixth one, AUG has dual functions. It codes for methionine and it also acts as initiator codon. The power of Perl for dealing with these features relies on the fact that Perl's powerful regular expression matching and string manipulation operators simplify the job of manipulating text, which often comes as DNA sequence. And Perl is component oriented, which means that Perl encourages people to write their software in small modules, either using Perl library modules or with the classic Unix tool oriented approach. External programs can easily be incorporated into a Perl script. A set of modules called BioPerl deals especially with biological counterparts. BioPerl is a collection of Perl modules that facilitate the development of Perl scripts for bioinformatics application. It has played an integral role in the Human Genome Project. The interpreter need not require the declarations of the function prototypes and data types in advance. The debugger is also very effective that it generates an error when an undefined function is used. Above all, it is very easy to write and fast to develop in. Using hashes for genetic code. If we use a hash for this translation, for each codon key, the amino acid value is returned. Here is the subroutine to translate a DNA to an amino acid using hash lookup. The subroutine is called codon to AA. It is a subroutine to translate a DNA 3-character codon to an amino acid using hash lookup. First of all, the arguments are collected and initialized to the variable codon. Now, the translating the codon to uppercase codon using $codon equal to UC $codon. And the genetic code is a hash which is initialized with TCA for S, that is serine, TCC for serine, TCG for serine, TCT for serine and TTC for phenylalanine. Here also the arguments are collected and initialized to the variable codon. Then the codon is translated to the corresponding uppercase codon as $codon equal to UC $codon. Then the genetic code hash is initialized with the 3-letter codon as its key and the first letter of the amino acid as its value. Here there is a number of initializations from serine to glycine. And then an if statement is used to check whether a codon exists in the genetic code, and if it is there, it will return the corresponding codon. Else an error message is printed. Here the subroutine initializes a hash with codon as keys and the amino acids as their value, and then performs a single lookup of its single argument in the hash. The keys are 64, each representing one codon. The exist function used here returns true if the key $codon exists in the hash. It is equivalent to the else statement in the 2 previous versions of the subroutine. This subroutine works on lowercase DNA and uppercase DNA. This is done by first translating the incoming argument into uppercase to match the data in the hash genetic code hash. Here there are 2 important things to notice. A regular expression can't be given as a key to a hash. So, the translation must be done first to make it a simple scalar value. The other is that character classes don't work in the keys for hashes. So, each of the 64 codons should be specified individually. Wrapping the code in a subroutine has some advantages. The subroutine does a little bit of error checking for non-existent keys. Additionally, if the entire source code for codon translation is written in the subroutine, it is easy to accomplish a new way of translation or to do lookups from a database or a DBM file. Then changing the internals of this one subroutine is enough to do the entire task. As long as the subroutine takes one codon as an argument and returns a 1-character amino acid That is, the interface to the subroutine remains the same. No need to worry about the accomplishment of task. Hence, the subroutine acts as a black box, which is one of the significant benefit of modularization of programs with subroutines. The advantage of using hash is that they are fast. Declaring the whole hash as and when the subroutine is called makes the program slow, and hence declare the genetic code hash only once as a global variable.